So today we will discuss an important topic, an interesting topic rather, related to bacteria. We will study how bacteria can be used as microbots or microrobots for the treatment of cancer. But first, I hope you guys have studied bacteria in your junior classes. It is not unknown that bacteria have been used as a host in genetic engineering for ages. But have you ever wondered, what if we can use bacteria as robots? Scientists have now made this possible. Let us see how. So first we will see a brief history on to the events that have ultimately happened before the development of bacteria as robots. So two independent researches have been carried out that have ultimately led to the development of nowadays bacterial microbots for treatment of cancer. So the first group of researchers focused on the development of bacterial therapies for cancer, and another group of researchers focused on the development of nanorobots for the treatment of cancer. So these independent research events had been carried out almost simultaneously that have ultimately developed today what we call as microbots or microrobots. Those consist of bacteria, consisting of bacteria that have ultimately developed to the therapies of cancer to a large extent. Let us see. So if we concentrate on the first group of researchers, so it was scientist William Coley who first carried out a lot of a mixture of concoctions of bacteria, and they infused into patients, okay, to treat various types of cancer during the year around 1890. So after some years, Bacillus calmette-guerin, this BCG attenuated tuberculosis bacterium, was also used as a vaccine strain for the development of bladder cancer during the time 1970s to 1980s. Okay, so after this, an attenuated strain of Salmonella typhimurium was also used to target tumors and inhibit their growth in mice. So, and after that, phase 1 clinical trial of this experiment was also carried out. Thereafter, numerous groups have established on further genetic engineering strains of E. coli and Salmonella, which have also showed the improved tumor targeting and drug delivery in vitro. So while this group of researchers on bacterial therapy for cancer was carrying out, another group of researchers were also carried out on development of nanorobots for cancer. So researchers developed radioactive gold nanoparticles for the development of liver cancer and sarcoma during the year 1954. Thereafter, they also developed liposome, and it was shown how liposome could also be studied with drugs for the treatment of cancer therapy. Thereafter, during the year around 1990, it was also shown that polylactic acid can be used for drug delivery for cancer therapy, and ultimately in the year 1995, the new complex was developed called as a liposomal nanoshell. So this liposomal nanoshell was developed, which incorporated chemotherapeutic drug doxorubicin, and this is the world's first approved nano-based medicine that was used in cancer therapy. So you can see these two simultaneous projects had been carried out almost towards the same year, and ultimately after 2005, so these two projects were combined into the development of a single project which we call as bacterial robots or bacterial microbots. So these bacterial microbots comprised of both the bacteriotherapy. Okay, so this comprises of both the bacteriotherapy as well as the nanorobotic therapy for the development of possible treatments of cancer. Okay, so it comprises of both the bacteriotherapy as well as the nanorobotic therapy. So it is actually a combination of the two therapies that we have seen. Before the development of bacterial robots or bacterial microbots, these two therapies were independent. Nowadays, scientists have clubbed these two independent researches and have combined them into one called as the bacterial robots. Okay, so and scientists also, these are researches are being carried out on how we can use more efficiently bacterial robots for the treatment of cancer. So now this is the most recent experiment or the most recent discoveries that have been made on the treatment of cancer. So this can be, if properly used, can have precise targeting of to tumor tissues and proper drug delivery. So nanotechnology has made bacterial microbots possible, although precise navigation to the target site is still a challenge. So this is one of the still major challenges, but hopefully we will overcome it someday, because these bacterial microbots have a great efficiency. Okay, if we can properly use them for proper targeting and drug delivery, so they can prove to be a huge blessing to science. So researchers have now developed a bacterial microbot for penetration into the body. So this microbot is loaded with drugs and they are targeted to the cancerous tissue to stop its proliferation. So let us now see how we can genetically manipulate bacteria so that we can use them as microbots. So there are mainly three types of mechanisms. So this is the first mechanism that we have. This is the second mechanism that we have. Okay, and this is the another one mechanism that we have. So there are mainly three types of mechanism by which we can genetically manipulate bacteria and use them as microbots. So first, what we will do, we can use live bacteria carrying virulent structures. Okay, so this is the virulent structure, and these virulent structures will ultimately result in death of the cancerous tissue by secretion of certain toxins. Okay, so how will they bring the death of cancerous tissue? By secretion of certain toxins. So in this way, this is one way in which we can also bring about the death of the target tumor tissue. There is another way by which we can modify the genome. So this is by genetic engineering. We can introduce the new form of DNA, okay, into the bacterial DNA, and we can create recombinant DNA, okay, and this recombinant DNA, if properly used, okay, if they are translated and transcribed, they will code for the synthesis of certain proteins that will ultimately prove to be harmful for the cancerous tissue, and they will bring about the death of the cancerous tissue. So by creating genetically modified DNA, so that means we are creating genetically modified DNA, that means we are using it as a host. Okay, so and we are from this DNA the transcription and translation will happen, that means gene expression will happen, and it will ultimately lead to the synthesis of toxins or the harmful chemicals which will ultimately result in the death of the tumor tissue. Okay, by gene expression from this rDNA, toxins will be produced, okay, which are mostly proteinaceous in nature. So there is another third group by which we can use bacteria as microbots, by using certain bacteria which will produce some cytotoxic enzymes. Okay, so by using certain bacteria which will produce cytotoxic enzymes, and these cytotoxic enzymes, cytotoxic means this will lead to the death of the mostly the cytoplasm of the tumor tissue, and then it will bring about the death of the cancerous tissue. Okay, so these are mainly the three mechanisms by which we can genetically modify the bacteria, and later we can use them as bacterial microbots to properly target to the tumor tissue and bring about the death of the cancerous tissue. So and this is one way. There is another way by which we can also load the bacteria with certain kind of anti-cancerous drugs, so that after the bacteria reach the proper target tumor tissue, they can they can release those drugs, and those anti-cancerous drugs will bring about the death of the cancerous tissue. So this is the mechanism by which we can load the drugs. So first mechanism is by surface loading of the drugs. That means drugs can be loaded into the bacteria by three methods: either by adsorption, either by electrostatic interaction, so or by covalent binding. So these three mechanisms, the proper anti-cancerous drug can be loaded into the bacteria, and later the drugs will be internalized into the bacteria. So here you can see, if you cut it vertically, you will see, and if you cut open it vertically, you will see inside these are the loaded drugs into the bacteria. Okay, and thereafter, on reaching the target tissue, these bacteria can release the anti-cancerous drugs by more or less 3 or 4 methods. These are chemical, biochemical, enzymatic degradation, okay, or by physical methods. So these 4 are the main methods by which the bacteria can release the anti-cancerous drugs. So first we will surface load the drugs into the bacteria. Okay, first we will surface load the drugs into the bacteria, then we will introduce it into the human body. While introducing it into the human body, the drugs will be internalized by the bacteria, and on reaching the target tissue, these drugs will be released by the bacteria. Okay, so and these anti-cancerous drugs will bring about the degradation of the cancer tissue. So now scientists observe many bacteria have the potential to selectively grow within solid tumors. Which bacteria have the potential, we will discuss next, but let us see. But one question that needs to be answered, that how can bacteria grow within tumors? So in the previous slides we have seen that bacteria can be targeted to the tumor tissue. Either they can be genetically modified, or they can be loaded with anti-cancerous drugs, and they can be targeted to the tumor tissue for destroying the tumor tissue. But it has also been seen that some bacteria also grow within the tumor tissue. So how do they grow within the tumors? How do they get the proper nutrients and everything for their growth and proliferation in the tumor itself? Reduced immune surveillance in the tumor's hypoxic and acidic environment provides anaerobic bacteria with a safe haven to grow and thrive there. So what happens? There is very much reduced immune surveillance in the tumor tissue, because because of this reduced immune surveillance and hypoxic condition. Hypoxic condition means there is almost no or very much less oxygen in the tumor tissue, and these conditions, reduced immune surveillance, hypoxic condition, and acidic condition, these three conditions provide the ideal situation, ideal conditions for bacteria, anaerobic bacteria to grow. One example of anaerobic bacteria that can grow within such conditions in the human tissue is the bacteria called as the Clostridia. Okay, so this Clostridia bacteria can grow within this is an obligate anaerobic bacteria that can grow within such conditions where you have reduced immune surveillance, where you have hypoxic conditions, and you have acidic conditions. So this is Clostridia is one such obligate anaerobic bacteria that can grow under such conditions. Growth of bacteria in tumors ultimately leads to immune cell infiltration, leading to anti-cancerous responses. So before the growth of bacteria, that there were reduced immune surveillance, there were the hypoxic conditions, there were acidic conditions. Okay, but after the growth of bacteria, there were the immune cell infiltrations, and that ultimately lead to anti-cancerous responses. Now, which immune cells are particularly important or involved in this anti-cancer responses on the growth of bacteria? These immune cells mostly involve CD8 plus T cells. Okay, here we have CD8 plus T cells. Then after CD8 plus T cells, we have macrophages. NK cells and dendritic cells. So these are the kind of immune cells which can bring about immune cell infiltration and anti-cancerous effects. So these are the mostly types of cells involved in the immune responses to cancers. Okay, so these only happen after the development of cancer, after the development of the growth of bacteria. So CD8 plus T cells, dendritic cells, macrophages, and NK cells. So these are mostly the four types of cells which can bring about such anti-cancerous immune responses. Okay. So this point that I have pointed out, this reduced immunosurveillance, so these points favor the growth of bacteria, and these points favor the growth of bacteria. Okay, and these happen before the growth of bacteria. Okay, so they favor the bacterial growth. So once the bacteria grows, immediately immune cell infiltration occurs, and ultimately anti-cancerous responses happen within the body that ultimately lead to the CD8 plus T cell activation, macrophages, dendritic cells, NK cell activation, and that ultimately bring about the tumor death of the tumor tissue. Okay, so these are mainly the conditions which can bring about the bacterial growth, and these happen after bacterial growth. Okay, after once bacterial growth occurs, you have the immune cell activation and immune cell infiltration. So we are very in a nurture stage. We are not at all in a very advanced stage. We are on a very basic stage. So because of that, researchers are now selectively trying to develop this technique more, and they are trying to selectively target and grow bacteria within the tissue so that automatic immune cell infiltration take place and anti-cancer response in place. So here, if we can grow the bacteria within the tissue, okay, so this is very important. If we can grow the bacteria within tissue, so if you can grow the bacteria within the tumor tissue, you do not need to genetically modify you do not need to genetically modify the bacteria, okay, and introduce into the body, or you do not need to load the bacteria with any sort of anti-cancerous drugs, because this is happening within the body. You are just providing the conditions which will encourage the development and the growth of bacteria within a tumor tissue. So once that happens, automatically your immune responses will be increased, and more amount of CD8 plus T cells, macrophages, dendritic cells will accumulate, and then they will be only responsible for bringing about the death of the tumor tissue. You do not have to do any kind of extra thing to bring about the death of the cancer tissue. Okay, you do not have to genetically modify the bacteria, or you do not have to do precise drug delivery. Okay, you do not have to load the bacteria with any anti-cancerous drugs. You do not need to do anything, but just you need to provide the ideal conditions, or you just to from outside you have to manipulate the conditions somehow or the other, so that the bacteria starts growing within a tumor tissue. So once the bacteria starts growing, automatically after a certain period of time your immune responses will increase, and then your cancer tumor tissue will be automatically destroyed. So you don't have to do any sort of genetic manipulation. You do not have to load the bacteria with anti-cancerous drugs, and then you do not have to introduce it into human body. So these stages are not needed at all. Just provide the ideal conditions for the growth of the bacteria, and the rest will be done automatically. So engineered bacteria, so we can also now let us see different methods of bacteriotherapy in cancer. So let us see the different methods by which we can bring about the treatment of cancer by using bacteriotherapy. So the first method is bacterial colonization and proliferation in and around the tumor tissue, resulting in death of the tumor tissue. So this is the tumor tissue that you can see, and here bacteria have colonized. Okay, so these are the bacteria that have colonized, and this bacteria after colonization secretes certain amount of toxins. So okay, these are the toxins that are secreted. As you can see, these purple portions, these are the toxins that are secreted, and these toxins will ultimately result in the death of the tumor tissue. There is another method of bacteriotherapy in cancer, is that you can introduce the bacteria into the body. The bacteria will bind to the complementary receptors present on the surface of the tumor tissue. So here you can see I have marked the receptor to which this bacteria is binding, and after binding, this bacteria will secrete certain amount of cytotoxic compounds which will ultimately result in the death of the cancer cells. Okay, so this is the first method. Either the bacteria can bring about the death of the cancer cell by this method, or they can bring about the death of the cancer cell by this method. Okay, so there is another method. Okay, so by this method, the bacteria, we can genetically modify the bacteria, as I have already told before. That can be used as a vector. Bacteria can be used as a vector, and by the rDNA or the recombinant DNA, the bacteria, that the recombinant DNA will produce certain amount of anti-cancerous agents that will ultimately bring about the death of the cancer cells. So you have a bacteria. Suppose you have a bacteria here, and you have introduced a recombinant DNA into this. Introduced a recombinant DNA into this bacteria. Okay, so this is that was a portion of the original vector, and this is the portion of the new DNA that you have introduced. So this is totally the recombinant DNA. So after this recombinant DNA, what will happen? On gene expression, it will produce some anti-cancerous agents. Okay, and those anti-cancerous agents will ultimately bring about the death of the cancer cells. So this is a technique of genetic engineering also, by which we can also bring about the death of the cancer cells. Okay, so there are a lot of methods. You can choose either of the methods to bring about the death of the cancer cells. There is another method. The last method is by triggering immune responses. So certain bacteria can on the precise targeting to the tumor tissue, they can activate the immune cells like CD8 plus cells, NK cells, they are dendritic cells, and those immune cells will ultimately bring about the cell death. So these are the 4 kinds of methods of bacteriotherapy by which cancer can be treated. Okay, so now let us see what happens after your goal is achieved, that is after the precise targeting and destruction of the cancer cells is done. Okay, so what will happen? Your after the bacteria destroys the cancer cells, the bacteria remains in that place. So you have to now remove or destroy the bacteria also, because now if you don't remove the bacteria, then that bacteria can also be harmful or somehow bring about some that bacteria may cause some disease symptoms. So what you will do after the precise targeting or the unloading of the proper anti-cancerous drugs, or after the destruction of the cancer cells, so once your goal is achieved, you will introduce some antibiotic drug molecule for the killing treated bacteria with antibiotics after tumor suppression. So after tumor suppression, so once the tumor is suppressed by the bacteria, so you have to kill that bacteria also, so that you remain free from any kind of other diseases that might be caused from this bacteria. Okay, so you kill this bacteria also by introduction of some antibiotic drug molecule, or by introduction of some antibody molecule which will opsonize this bacteria and remove it. So this process only happens after your target is achieved, that is after the destruction of the tumor tissue or the tumor suppression occurs by the bacteria. So what happens? First you introduce the bacteria, and the bacteria destroys the target tumor tissue by any one of the four following methods. Okay, any one of the four methods. Either it can synthesize bacterial toxins, or it can destroy the cancer cells by producing cytotoxic compounds, or it can can be used as a vector to synthesize some anti-cancerous agents which will bring about tumor suppression, or it can bring about immunostimulation. So after your target is achieved, after your tumor is suppressed, then you have to remove the bacteria that you have used for tumor suppression also, so that your body doesn't suffer from any sort of other diseases. Okay, so how we will do, you can do it by either of the two methods, by introducing any antibiotic which will destroy that bacteria. Okay, or you can use some antibody which will opsonize and destroy the bacteria. So after your job is done, you will also destroy the bacteria also. So this is another mechanism where you will see in detail how bacterial microbot action happens. So the first method is that after you introduce the bacterial microbot into the So this is another slide where you will see how the mechanism is achieved by the bacterial microbot. So after you introduce the bacterial microbot into the human tissue, okay, so it penetrates the veins and the capillaries. This is the first step. It penetrates the veins and the capillaries. You can see these greenish portions within the veins and the capillaries. These are the bacteria that have penetrated the veins and the capillaries. Okay, so after penetrating the veins and the capillaries, the ligands attached to the surface of the bacteria. So ligands attached artificially to the surface of the bacteria binds to complementary receptors on the surface of the tumor tissue. Okay, these ligands are attached for the proper binding to the receptors of the on the surface of the tumor tissue. So after penetration of the veins and the capillaries, what happens? They reach the target tumor tissue, and they bind the tumor target surface. They bind the receptors on the target tumor tissue with the help of the ligands which are attached on them, and then they can bring about the death of the tumor tissue by either immunostimulation, or by production of some toxic or cytotoxic compounds, or by some anti-cancerous agents. So either of the following methods they can do that. Okay, after the proper binding to the receptor on the surface of the tumor tissue. So first is vein and capillary penetration by the bacteria. So these greenish portions within the brains, you can see these are the bacteria. So after vein and capillary penetration, they reach to the target tissue where they bind to the surface of the receptor with the help of the ligands that are attached on them. So binding to the receptor by means of the ligands that are attached. So these are the ligands, and here you have the receptors that are receptors that are present on the surface of the tumor tissue. So binding of the ligands to the receptors on the surface of the immune cell, and then they can bring about either immunostimulation or the production, or they can produce some cytotoxic compounds which will ultimately bring about the death of the tumor tissue. Okay, so now one question arises, what is the stimulus? Okay, or how you will guide the bacteria throughout so that bacterial microbots reach the proper tissue. Okay, how we will guide them throughout their own journey. Okay, so whole journey, how we will guide them. Okay, because what happens, you need to have a stimulus, or you need to show the bacteria its path so that it reaches the proper tissue and not any other tissue. Okay, so how you will do that? You can use three different kinds of stimulus. You can use either light, so the journey of the bacterial microbots to the target tumor tissue can be either light guided or magnetic field guided. Okay, so you can provide the suitable magnetic field of suitable frequency and the bacterial microbot will reach to the target tissue, or they can be ultrasound guided. So these are the mainly the three different stimuli which you can provide to the bacterial microbot so that they reach their target tissue and not any other tissue. Now let us see some examples of bacteria which can be used as bacterial microbots. Okay, so two virulent genes, these are MSB and PUR1, were deleted from Salmonella typhimurium. Okay, and then they were after deletion of these two virulent genes. So remember, these two genes were virulent genes, MSBP and PURI. So these two genes were virulent genes. Why they have been removed? Because they need to be introduced within the human body, right? So if they wouldn't have been removed, then they could have caused some disease symptoms within the human body. So that's why they have been removed, and after removal of these virulent genes, Salmonella typhimurium were loaded with therapeutics for cancer treatment. Okay, then encapsulated in biocompatible alginate molecules. So after loading of the proper drugs, they were encapsulated in biocompatible alginate molecules and then delivered to the target tissue. So four steps happened. First is virulent gene removal. Okay, then loading with the anti-cancerous drugs. Okay, loading with the drugs, then encapsulation in biocompatible alginate molecules, and then final drug delivery and unloading of the drug. So these four steps happened that ultimately brought about the death of the tumor tissue. Next, another bacteria we have Listeria monocytogenes. So these bacteria had also been used as bacterial microbes for the treatment of cancer. So this bacteria synthesize Listeriolysin O that can cause bacterial survival by preventing phagolysosome formation in host cells leading to immune cell activation. So what happens, this Listeriolysin O, okay, this Listeriolysin O causes bacterial survival. That means it helps in the survival of the bacterial Listeria by preventing phagolysosome formation in the host cells. So if you introduce this bacteria into the body, it will synthesize this Listeriolysin O that will ultimately prevent the formation of phagolysosome. Okay, so within the human body, because phagolysosome will destroy the bacteria, so it will prevent its formation, and it will grow within the tumor tissue. And ultimately, what I have explained before, that when the bacteria will grow within the tumor tissue, immune cells will be activated like CD8 plus T cells, NK cells, dendritic cells, and ultimately those immune cells will ultimately bring about the tumor suppression. So this is another method. Then we have the bacteria Bifidobacterium. So this bacterium can disrupt toxic fecal enzymes, thereby exerting anti-cancerous effects. So these fecal enzymes are important for tumor growth. So they can disrupt this toxic fecal enzymes and they can exert their anti-cancerous effects in that way. So another bacterium, so this I have already discussed, this Clostridium bacterium is actually an obligate anaerobic bacterium. So this the obligate anaerobic bacterium can partially lyse cancer cells and convert inactive prodrugs into cytotoxic drugs at the target site. So this is one of the main functions, and this is one of the main bacteria that have been used as bacterial microbots. Okay, now why this bacteria is of considerable choice is that I have already told you that within the tumor tissue, the environment is extremely hypoxic. That means very less the oxygen, and extremely acidic. So this Clostridium bacterium is one such anaerobic bacterium which has this ideal condition for its growth. Okay, so that condition is very much ideal. So the condition that you will find within the tumor tissue, the hypoxic and acidic conditions, that conditions are very much ideal for the growth of such kinds of bacteria. So that's why Clostridium bacterium is of very much considerable choice. And another point why it is also of considerable choice is that it can convert some inactive prodrugs into cytotoxic drugs also within the human body, and those cytotoxic drugs can bring about the tumor suppression. So here in the figure you can see these Clostridium bacteria, can reach the solid tumors by means of aerotaxis. Aerotaxis means oxygen is used as a stimuli for that taxis. Okay, so oxygen, aero means oxygen, so they can sense the deficiency of oxygen, because within the tumor, I have already told you, the tumor environment is very much hypoxic, so they can sense the deficiency of oxygen, or they can sense the changes in the level of oxygen, and they can reach the solid tumor, and they can bring about the immune cell activation, and then T cells, macrophages, neutrophils, NK cells. So these are the four kinds of immune cells which will become activated, and they will start accumulating, and then these immune cells will produce some tumor chemokines that will ultimately bring about the death of the tumor tissue. Next we have Vaccinia. This bacteria can also exert its anti-tumor, anti-inflammatory functions by secretion of some exopolysaccharides. Okay, so another we have E. coli has also been used for the treatment of cancer as bacterial microbots. So this slide is very important, where you can see Salmonella bacteria has been used. Listeria monocytogenes can also exert its anti-cancerous effects by production of listeriolysin. Bifidobacterium can also provide its anti-cancerous effect by disrupting of certain toxifical enzymes. Clostridium bacterium can also provide some anti-cancerous effects by converting some inactive products into active cytotoxic drugs, and Vaccinia can also provide anti-cancerous effect by secretion of some exopolysaccharides. Along with that, E. coli, non-pathogenic E. coli, that means you remove the pathogenic portions from the E. coli and make it non-pathogenic. They have also been programmably lysed in the tumor environment, and by programmable lysin, they can release their loaded drugs into the tumor environment and bring about tumor suppression. Now we will see some common bacteria which have the potential to be used for anti-cancer therapy. Okay, so the first bacteria that we will see, so these are those bacteria which have the potential, which can be used in future for cancer therapy, and most probably they can be used as bacterial microbots also. So Bacillus licheniformis, this bacterium produces the peptides iodoglobamides A and B. Okay, and it has been shown that this peptide is effective against human gastric, stomach, and lung cancer cell lines. So if we can manipulate this bacteria somehow genetically or in vivo, then we will see this peptide can bring about effective treatment for human gastric cancer, human stomach cancer, or human lung cancer. Next we have Nocardiopsis. This bacterium also can bring about some anti-cancerous effects. So by production of this peptide leucentamines, and it has been seen that this peptide is effective against human colon carcinoma cell line. Next we have the bacterium Enterococcus. So this bacterium produces the peptide NTAP, and it can bring about anti-cancerous effects against adenocarcinoma cell lines. And how it does is by interfering cell proliferation and inducing apoptosis. So by these two mechanisms, interfering with the cell proliferation and inducing apoptosis, it can bring about the tumor suppression in case of human adenocarcinoma cell lines. Another bacterium we have, this is Streptococcus pneumoniae. Okay, so this bacterium produces the peptide PEP27, and it is effective against AML2, HL60, and MCF7 cancer cell lines. And the last bacterium, this bacterium is very much famous, and many researches have also been carried out on this bacterium. This is Helicobacter pylori. This bacterium produces the Helicobacter pylori ribosomal protein. So this ribosomal protein is very much important. This protein is produced by this bacteria, and it has been seen that it is effective against Hep G2, BGC823 human cancer cell lines, and it can bring about tumor suppression by activating caspases. Caspases are involved in apoptosis. Okay, so their caspases are proteins involved in apoptosis, so they can in a way induce apoptosis by activating caspases, and they can induce the formation of ROS. ROS means reactive oxygen species. Okay, so like free radicals, superoxides, etc., and inhibiting the cell cycle against these kinds of cancer cell lines.